Hi friends, welcome back to my channel. So if you're new to my channel, please kindly subscribe, like and share it with your friends also, so that it would be helpful for them also. Okay, so without any late, let's start with the class. Okay, so today we are going to discuss from here. The nucleus contains the cell's DNA. The nucleus, the eukaryotic cell's most conspicuous organelle, is the repository of its genetic information. So if we see the nucleus, this nucleus is present in the eukaryotic cell, where most conspicuous organelles are present along with the genetic information. Okay, this information is encoded in the base sequence of DNA molecules that form the discrete number of chromosomes characteristics of each species. So the information which is encoded in the base sequence of DNA molecule, it is going to form in the discrete number of chromosomes. So they are all formed in a number of chromosomes. Okay. So the base sequence of DNA molecules are formed in the discrete number of chromosomes where the species is characterized. Okay. The chromosomes consist of chromatin, a complex of DNA and protein. So chromosome consists of a chromatin, okay, a complex of DNA and protein is present in it. Okay, the amount of genetic information carried by eukaryotes is enormous. For example, a human cell has over 700 times the DNA of E. coli. So if we see the amount of genetic information which is carried by eukaryotic cell is more enormous. Okay, example, if we see the human cell, over 700 times the DNA of E. coli is present in it. Okay, in the terms commonly associated with computer memories, the genome genetic complement in each human cell specifies around 800 megabytes of information, about 200 times the information content of this text. So the genome which is giving the text in each human cell is specifying around 800 megabytes. Okay, so it is giving the information around 800 megabytes, where this 800 megabytes is around 200 times more than the content. Okay, within the nucleus, the genetic information encoded by the DNA is transcribed into molecules of RNA. So within the nucleus, there is a genetic information which is encoded by the DNA, and DNA is transcribed into the molecules of RNA. Okay, which after extensive processing are transported to the cytoplasm. So after the DNA, the genetic information which is encoding to the DNA, then it is transcribed to the molecules of RNA. So these molecules of RNA are then extensively processed and then transferred or transported to the cytoplasm. Okay. In eukaryotes, the cell content exclusive of the nucleus, where they direct the ribosomal synthesis of the proteins. So if we see in eukaryotes, the cell contents exclusive of the nucleus, right, where they direct the ribosomal synthesis of the protein. So this ribosomal synthesis of proteins are done in the cytoplasm itself. So in the cytoplasm, there is ribosome present in the eukaryotes. Okay, so ribosomes are present in the cytoplasm, where the ribosomal synthesis of protein is done. Okay, the nuclear envelope consists of a double membrane that is perforated by numerous approximate 90 Armstrong wide pores that regulate the flow of proteins and RNA between the nucleus and the cytoplasm. So the nuclear envelope is of double membrane, right, and it is perforated by numerous approximate 90 Armstrong wide pores. So it is having a wide pores which is approximate 90 Armstrong. Okay, so they are going to regulate the flow of proteins and RNA between the nucleus and the cytoplasm. Right? The nucleus of most eukaryotic cells contains at least one dark staining body known as the nucleolus, which is the site of the ribosomal assembly. So the nucleus which is present in the eukaryotic cell, it contains at least one dark staining body known as a nucleolus. right? So the nucleus is having a nucleolus where the site of ribosomal assembly is done. Okay, it contains chromosomal segments bearing multiple copies of genes specifying ribosomal RNA. So this nucleolus is containing a chromosomal segments bearing multiple copies of genes specifying the ribosomal RNA. These genes are transcribed in the nucleolus. So the genes are transcribed in the nucleolus and the resulting RNA is combined with ribosomal proteins that have been imported from their site of synthesis in the cytosol. So these genes are transcribed in the nucleus and it is going to result in the RNA, right? So this RNA is combined with the ribosomal proteins and they are imported from their site of synthesis to the cytosol. Okay, cytoplasm exclusive of its membrane bound organelles. The resulting immature ribosomes are then exposed to the cytosol where their assembly is completed. So at last resulting the immature ribosomes are then exported to the cytosol where their assembly is completed. Okay, thus protein synthesis occurs almost entirely in the cytoplasm. So if we see the protein synthesis, it is almost entirely done in the cytosol. Okay, cytosol or cytoplasm, right? Next, we will be seeing about the endoplasmic reticulum and the Golgi apparatus function to modify membrane bound and secretory proteins. Now we will be seeing about this topic, right? The most extensive membranes in the cell, which was discovered in 1945 by Keith Porter, form a labyrinthine compartment named the endoplasmic reticulum. So there is a most extensive membrane in the cell which is discovered in 1945 by Keith Porter, scientist, right? So Keith Porter is a scientist in 1945. He discovered the most extensive membrane in the cell. Okay. So he found that the membrane is formed a labyrinthine compartment named the endoplasmic reticulum. So this endoplasmic reticulum formed like a labyrinthine compartment. Okay, a large portion of this organelle called the rough endoplasmic reticulum. So in this endoplasmic reticulum, there is a large portion of organelle called rough endoplasmic reticulum is studded with ribosomes that are engaged in the synthesis of proteins that are either membrane bound or destined for secretions. So this rough endoplasmic reticulum is studded with the ribosomes. That's why it is known as a rough endoplasmic reticulum. Okay. So there are small ribosomes which are present on the rough endoplasmic reticulum. Right. They are engaged in the synthesis of proteins that are either membrane bound or destined for the secretions. There is another endoplasmic reticulum, the smooth endoplasmic reticulum, which is devoid of ribosomes, is the site of lipid synthesis. So if we see the smooth endoplasmic reticulum, it is devoid of ribosomes. So it does not contain any ribosome. Okay. So it is the site where the lipid synthesis is done. Okay. Many of the product synthesized in the endoplasmic reticulum are eventually transported to the Golgi apparatus named after Camillo Golgi, who first discovered it in 1898. So many products synthesized in this endoplasmic reticulum and are eventually transported to the Golgi apparatus. So this Golgi apparatus was discovered by the scientist Camillo Golgi. Okay, Camillo Golgi, scientist who discovered this Golgi apparatus for the first time in 1898. Okay, a stack of flattened membranous sacs in which these products are further processed. So there is a stack of flattened membranous sacs in which these products are further processed. Okay. So we will be seeing about the mitochondria are the site of oxidative metabolism. The mitochondria, Greek mitos means thread, chondros means granules. So thread granules totally are the site of the cellular respiration, aerobic metabolism in almost all eukaryotes. So mitochondria is the site where the cell is respiring, right? So cellular respiration is done in almost all eukaryotes. So it's an aerobic metabolism, right? So to know about the mitochondria, mitos means it's a thread. Mito means thread, Chondria means granules. So granules like mitochondria are present. Okay. These cytoplasmic organelles, which are large enough to have discovered by 19th century cytologists, vary in their size and shape, but are often ellipsoidal with dimension of around 1 into 2 micrometers, much like a bacterium. So the cytoplasmic organelles, which are large enough, they have been discovered by 19th century itself, okay, by a cytologist, varying in their size and shape. They looks like a ellipsoidal with the dimensions of around 1 into 2 micrometers, like a bacterium. Okay. A eukaryotic cell typically contains on the order of 2000 mitochondria, which occupy roughly 1/5 of its total cell volume. So if we see this eukaryotic cell, typically it contains an order of 2000 mitochondria. Okay. So these mitochondria are occupying roughly 1/5 of its total cell volume, right? So the mitochondrion, as the electron microscopic studies of George Pallade and Fritjof Sorstrands first revealed has two membranes, a smooth outer membrane and a highly folded inner membrane, whose invaginations are termed cristae. So the mitochondria is further seen in the electron microscope studies. So for the first time, George Pallade and Fritjof Sorjostrand first revealed that this mitochondria is having two membranes, right? One is the smooth outer membrane, right? And the inside one is the highly folded inner membrane, known as a cristae. Okay. So the mitochondria will be looking like this. So this is the smooth outer membrane, right? So the another one which is inside, so it will be looking like a cristae. Okay, so it is looking like this. So this we will be calling as a cristae. Okay, this we will be calling it as a cristae or crest. Okay, thus the mitochondrion contains two compartments, the intermembrane space and the internal matrix space. So it is having a two compartments where the intermembrane space. So all this space we will be calling it as a intermembrane space. Okay, and then internal matrix space. So this is the internal matrix space. Okay, this is the internal matrix space, right? The enzyme that catalyzes the reactions of respiration are components of either the gel-like matrix or the inner mitochondrial membrane. So if we see the enzymes, they are catalyzing the reaction of a respiration. Okay, reaction of a respiration, and the compartments of either they looks like a gel-like matrix or the inner mitochondrial membrane, right? These enzymes couple the energy producing oxidation of nutrients to the energy requiring synthesis of adenosine triphosphate ATP. So the enzymes which are coupling the energy producing oxidation of nutrients to require energy, they are synthesizing with the help of a adenosine triphosphate ATP. So they are utilizing the energy, right? Adenosine triphosphate, after export to the rest of the cell, fuels its various energy consuming process. So after the ATP is utilized in the enzyme coupling reactions, the nutrients are requiring the synthesis, right? So after synthesizing, this adenosine triphosphate is after exported to the rest of the cell. Okay, then the fuel is going to consume the energy processes, right? Next, mitochondria are bacteria-like in more than size and shape. So if you see the mitochondria, they are bacteria-like in more than size and shape. Their matrix space contains mitochondrial specific DNA, RNA and ribosomes that participate in the synthesis of several mitochondrial components. So if we see the matrix space, it contains a mitochondrial specific DNA, RNA and ribosomes. They are going to participate in the synthesis of several mitochondrial components. Moreover, they are reproduced by binary fission, and the respiratory processes that they mediate bear a remarkable resemblance to those of modern aerobic bacteria. So this mitochondria is reproducing reproduced by a binary fission, and the respiratory processes that they mediate bear a remarkable resemblance of modern aerobic bacteria. So this mitochondria is reproducing by a binary fissions and a representative processes, right? These observations leads to the now widely accepted hypothesis campaigned by the Lynn-Margulis that mitochondria evolved from originally free-living gram-negative aerobic bacteria which form a symbiotic relationship with a primordial anaerobic eukaryotes. So here these observations led wide accepted hypothesis by Lynn-Margulis that mitochondria evolved from free-living gram-negative aerobic bacteria, which they are formed by a symbiotic relationships with the primordial anaerobic eukaryotes. Okay. The eukaryotes supplied nutrients consumed by the bacteria were presumably repaid several folds of the highly efficient oxidative metabolism that the bacteria conferred on the eukaryote. So eukaryotic supplied the nutrients consumed by the bacteria were presumably repaid several folds of highly efficient oxidative metabolism that the bacteria conferred on the eukaryote. Okay. This hypothesis is corroborated by the observation that the amoeba, Paleomyxa palustris, one of the few eukaryotes that lack mitochondria, permanently harbors aerobic bacteria in such a symbiotic relationship. So the hypothesis is corroborated by the observation of the amoeba. The amoeba name is Paleomyxa palustris, one of the few eukaryotes which is lacking the mitochondria permanently. Okay, harbors, and it is harboring the aerobic bacteria in such a symbiotic relationships. Okay, so okay friends, thanks for watching. If you like my video, please kindly like, share and subscribe it with your friends also. Okay, so in the next class we will be studying about the lysosomal and peroxisomes are containers of degradative enzymes. We will be continuing it in the next class. Okay, thanks for watching. Bye bye.